Let's try. I do have a.
classroom. Nuestra gratitud a todos. Hoy lo hacen a través de YouTube. Our gratitude to all the interested people who are connected today through the internet at Zoom. And of course, to those who today is on YouTube. De manera virtual y sin la presión del tiempo, nuestro agradecimiento también ya incluye a quienes en algún momento a futuro verán este evento, por supuesto, que será en la modalidad de video dentro del mundo de YouTube. Para entonces será histórico el constatar lo que hoy estaremos físicamente construyendo en forma presencial. Se trata nada más ni menos que una alianza, un trabajo de cooperación entre investigadores del Instituto Politécnico Nacional y de la Universidad de California, en torno a un tema de muchísima relevancia, yo diría de importancia vital. En el área de la salud, voy a hablar del corazón. Virtually and without the pressure of time, our gratitude already includes those who in the future will be seeing this event. Of course, it will be in the form of a YouTube video. By then, it will be studied in person. This is an alliance. I would say of vital importance in the area of health. We are talking about heart. Dr. Arash, extraordinario y muy destacado investigador de la Universidad de California, compartirá con nosotros su visión sobre la investigación actual en el sistema cardiovascular y abordará algunos de los avances que a la fecha se han logrado en este tema. Dr. Arash, extraordinary and outstanding researcher at the University of California, will share with us his view on research on cardiovascular systems and will address some of the advances that have been done in this area. Esta será una gran oportunidad para lograr un serio y detallado análisis del potencial que existe entre ambas instituciones, entre el Politécnico y la Universidad de California. Y sobre todo de la relevancia de trabajar en equipo en temas de interés común. This will be a great opportunity to achieve a serious and detailed analysis of the potential that exists between both institutions. I'm talking about the Instituto Politécnico Nacional and the University of California. We are very close. And above all, of the relevance of working together as a team on issues of common interest. Dr. Arash, Comunidad Politécnica, estamos seguros de que este es el inicio de un gran proyecto conjunto de mediano y largo plazo en el campo de la ciencia y sobre todo de relevante impacto en el campo de la salud. Sean todos ustedes bienvenidos. Dr. Arash, Polytechnic Community, we are sure that this is the beginning of a great medium and long-term joint project in the field of science, and above all, with a relevant impact in the field of health. Welcome, everyone. Doctora Tania Libertad Jacobo Estrada, the floor is yours. We will be a pleasure hearing some words about the Secretary of Postgraduate and Research of our Instituto Politécnico. Tania, will you please? Yeah. You can do it.
Okay, perfect. Thank you. First of all, I would like to apologize if my English is not so good. I can assure you I'm going to try my best to make the least number of mistakes in this very short speech, but your patience and your understanding deeply appreciated. Having said that, good morning, fellow members of the IPM community of this beautiful center, and particularly our guest of honor, Dr. Kheradvar. Unfortunately, the Secretary of Research and Graduate Studies, la doctora Laura Arreola, couldn't be here due to prior commitments, but she sent you her warmest regards. Thank you so much for this invitation. I am very glad to be here in this event, that it's gonna allow me to be witness in this new chapter in the history of this center, and of course, in the historic collaboration between the IPN and the University of California. I would like to thank Dr. Kheradvar for your visit and, of course, for your presentation, because without a doubt, it will ignite ideas and discussions about the ways in which we can collaborate further and contribute to the advancement in the solutions to important health issues worldwide. I'm sure that working side by side, we can have a better chance to find the solutions that we need. Now more than ever, it has become of clear importance, the clear importance of the scientific community to effectively address this type of issues. And now more than ever, the scientific community has shown its commitment to respond to the needs and demands of the society that it belongs to. I am sure that the community of the IPN is up to the challenge and what is expected from us. Also, I would like to applaud the academic community of this center for your commitment and devotion to do science of the highest end, and your willingness to exchange ideas and collaborate with colleagues of the most prestigious universities in the world, such as the UC Irvine. Finally, I would like to reaffirm that you have the full support of the Secretariat of Research and Graduate Studies to facilitate the development of research projects that may derive from this collaboration. I am sure that great things are ahead of us from this visit. Welcome once again, Dr. Kheradvar, and thank you for your attention.
Thank you, Dana. We are in the Computing Research Center, which is one of the best buildings and staff and community of our Instituto Politécnico Nacional. I might say it proudly. So let's have the opportunity to hear from our principal, the Dr. Francisco Irán Calvo Castro, who is in charge of this unit, academic unit and research unit. Dr. Francisco, the floor is yours.
Thank you very much. First of all, thank you all of you for coming here to listen to this very interesting talk, which Professor Kheradvar will start in a few minutes. I am sure that this interest of learning and further developing your knowledge and interacting with these kind of things will benefit all of us for growing as a center and developing solutions that finally will help everyone to have better solutions, in particular for health development. So I think that in particular, the joint contribution for the computing science areas that we develop here at the center will be in a very good combination with this knowledge that Professor Kheradvar brings to us today. So I see this as a very good, a very nice opportunity of having this kind of interaction. And I am sure that this will not be the last time that Professor will visit us, because also I have talked with him and he is very fond of Mexico. So I'm very happy to have him here, not only for visiting or having holidays and knowing our places we have to offer, but also the cultural, the knowledge, the research, the business that we are also able to offer. Well, this is a very nice opportunity, and I'm also very happy to be here with you, sharing this opportunity today with all of you. So I have no more words. So just to thank you very much to everyone here, you, and of course, Dr. Tania, thank you also very much for coming here, and also thank you very much to Dr. Laura Arreola, please send to her also our warmest regards. So this is all from my part. Thank you very much.
Thank you, Doctor. Let me say some words for the presentation of Dr. Arash Kheradvar. We have prepared a very small video. So I will ask to the three our guests, please, to go to the front in order that you can see the video. While they are moving, I might say what we have planned. First of all, we will see a presentation of the University of California campus Irvine. So where does this come? It comes from UC, University of California, Irvine, and then a small presentation of Dr. Arash Kheradvar. Thank you, Doctor, for being here. So are we ready for the video? Okay. In the first part, we will see what's University of California. Thank you.
Over 50 years ago, an extraordinary group of faculty, students, and staff arrived on the rolling hills of Irvine Ranch. They were innovators. They were visionaries. They were committed to creating one of the truly great centers of higher learning in the land. And they exceeded everyone's expectations. Today, it is our job to carry that pioneering spirit into the future. For our goal is simple, but profound. We generate knowledge and translate that knowledge into real-world impact. We enrich the minds and spirits of young people so they can develop to their full capacity. We enlighten our communities and point the way to a better future. A future where an elite education is not just for the elite, an education that is inspired by people from a variety of backgrounds and enhanced by the community around us. For each and every one of us has something to give. And when we have the courage to follow our convictions, together we can make a world of difference. Join us on our journey as we lead the way to a brighter future.
That's a very small and clever presentation of the University of California, Irvine. Now, we did a small work yesterday, Doctor, for him to have the presentation of our guest, Dr. Arash Kheradvar. Please.
The projects developed in this laboratory
No se alcanza.
Dr. Kheradvar has driven innovation. Dr. Kheradvar's research focuses on
¿No? Hagamos un pequeño ajuste.
developed in
Si me
this
permiten
laboratory.
tantito. Préstame el documento. Let me mention some additional notes from Dr. Kheradvar. Dr. Arash research focused on cardiovascular engineering with emphasis on cardiac mechanism, cardiac imaging, and the cardiovascular devices. Among the projects developed in his laboratory in the area of design and development of artificial heart valves, multi-scale modeling in congenital heart diseases, development of a 3D echoparticle imaging velocimetry system for the evaluation of the right ventricle, and the design of the new invasive, non-invasive indices for the quantitative evaluation of diastolic dysfunction. It's not so easy to pronounce many words you are not related. Dr. Arash has driven innovations in the field of heart valves and cardiac imaging, with more than 30 issues, use patents, and more than 60 international journal articles. Dr. Kheradvar has built a portfolio of successful products for the concept stage and led them to winning technology that are currently in preclinical stage and clinics. He leads diverse interdisciplinary groups of engineering, physicians, and residents to try the translation of scientific knowledge to the benefit of patients and society. Dr. Kheradvar is the author of 2 books. The first one, Principles of Heart Valve Engineering, and the second one, Vortex Formation in the Cardiovascular System. He is the member of the American Institute of Biological and Medical Engineering and the American Heart Association, Radiology and Interventional Cardiovascular and Cardiovascular Surgery and Anesthesia. Let's give a welcome to Dr. Arash.
Thank you very much. It's an honor to be here.
The stage, the internet, and many people that are today in Zoom and YouTube is with you. Thank you.
Sure. Thank you so much, everyone. It's an honor to be here. It's an honor to be here. Thanks so much for your hospitality and invitation. I'm very excited to be here. I genuinely love people of Mexico, and I really much believe in their capabilities and what they have done through the history and now. So I have always been excited to start a fruitful collaboration with Mexican scientists and engineers. So I'm very excited about this opportunity that is happening right now. So hopefully we can start some very fruitful collaboration in multiple lines of projects that eventually going to help the human health. I put together a presentation that's covering some of the areas that we are working in my lab, and this means to kind of show you what opportunities may arise from the collaboration between the 2 universities. So my talk is going to be about the trends, advancement, and challenges in cardiovascular engineering. And generally, as I said, I'm going over some of the projects that I have in the lab, and would love to later on talk to you in person about each of those that you do have some interest, so we can continue working on together. So in my lab, as I said, there are multiple projects, and they span from heart valve like structural heart engineering, which includes the heart valves, cardiac support devices.
Recording in progress.
And also cardiovascular imaging. So as you see, there are multiple projects that could be completely independent of each other, but every one of those are focused to improve the cardiovascular diseases, like the way that we treat or diagnose the cardiovascular disease. So, like, as I said, half of my lab is kind of focused on cardiovascular devices that are going to help the structural heart disease. And the other half are cardiac images, and there's some projects that are in the intersection of both devices and images. So I know that you have been very advanced in artificial intelligence and deep learning as I through my conversation with some of you when I was in California. So one of the line of research that I have been following, and that's mainly in collaboration with Professor Hamid Jafarkhani, who is also a chancellor professor at UC Irvine, is to take advantage of the advanced artificial intelligence and deep learning methods to be able to automatically segment the heart and diagnose a particular diseases for the heart. So we have been successful to develop algorithms that without the need of any physician or any scientist automatically can take the medical image data, like in this slide, mainly the cardiac MRI, and are able to fully characterize the right ventricle and left ventricle. One of these papers is currently a state-of-the-art paper, has over 500 citations. That's basically what, as we developed a hybrid method that takes advantage of the deep learning and deformable models, which I'm sure you're familiar with that. Next slide, please. We have also been using the generative adversarial networks, or GAN, which this is a new method, new technique, relatively new technique that has been using in computer science. We have been able to use those for defining like artificial cardiac images, and those artificial cardiac images can be used for training the networks, because as you know, for many of the diseases of the heart, we do not have so much data that requires for training the deep learning methods. So we come with this idea to generate synthetic data that are not possible to distinguish, or very difficult to be distinguished from real images through a specialized physician who are really used to do working with those images. Can you go for the next slide? So one of those is style GAN, which is style GAN is basically a generator that's built from the number of generative blocks, and it basically evolve into a network and ensuring the stability and minimize the mode collapses and capture the high-level features before low-level ones. And this is kind of like, it shows the way that it generates the images. So this is basically the faces, and this is not our work. This is just the show that the source is shown here. This is just show how the system works. And if you go to the next slide, you see two images, two cardiac MRI images, which one of them is real and one of them is fake, basically synthetically made, does not belong to any person, any real person. And then we developed that based on the style GAN in collaboration with Professor Jafar Khani's team. So this is basically the one on the right is real and the one in the left is synthetic. And these are very, very difficult even by the, to distinguish even by the best and the best of radiologists. Next slide, please. So we have been working in this area for quite a few years. This is one of the more recent studies that we published in Royal Society Interface. This is for the first time discussing deep learning methods for multi-label segmentation of echocardiograms. Echocardiograms are basically coming out of the ultrasound machine, and those are the ones that when you go to a physician, if they want to look into your heart, they put a probe on your chest and they look into the heart. That's usually considered the simplest and the cheapest way of studying the heart. But obviously, the way that it works is that usually an echo technician takes the images and then calculates some indices and then they provide it to the physicians. But if you go to 5 cardiologists and they basically scan your heart, they give you 5 different sets of measurements. So we try to come with a generalizable that consider the differences between the experts. Technique learns from multiple different experts and put together all the differences. Our goal is for the platform to provide a measurement that it's a standard deviation or error. If you look at it among the 5 other leaders in that field, the standard deviation would be minimal. Because as you see, as you know, the ground truth for these things are not unique. So like if you ask 5 Nobel laureate on this thing, if they look at it, all of them are right. And then if you look into their results, you will see that there is slight differences. So our system aims to basically capture the one that's a consensus of every medical experts on this field. Next slide, please. So that's basically a lot of work that we have done in the segmentation and analyzing the heart automatically through computers without the need of any physicians or engineers. And these are some of the works that I showed you has been published, and obviously we are still actively working on that. We recently got a grant from American Heart Association, and with the help of them and in collaborating with them, we are hoping to develop a platform that's cloud-based, and it's devoted to the diagnosing of the congenital heart disease for the children. And the beauty of this platform that we are developing is that anybody from anywhere in the world, like even in the places that you rarely have a physician there, as far as they do have an MRI system that they can capture the data, they can upload that data for free to our platform, and immediately they receive the analysis and the segmentation for that data set. And that's a very important step in helping these children. Assume that in many areas that we do not have the subspecialized physician who are expert in congenital heart disease. And analyzing congenital heart disease is the most difficult among the areas in radiology because of every heart is very different and it requires a lot of expertise and experience to be able to diagnose those. So I stop on that project and then go to a next project that I have been working on for over 15 years. And that's in fluid dynamics. It's called particle image velocimetry. So this is not a new technique, it's been done since 70s. So when you do have a particle flow or flow that's seeded with some particles, if you do have at the image of the particle flow in a time t, and then if you do have the same image in the next delta t, then you will be able to capture the velocity field in the flow. And that's very important in the fluid dynamics. And there are, as I said, since like an early 70s, they have been working on that. Most of the time in fluid dynamics, they use laser sheet to do that. Basically, the velocimetry techniques are based on like mathematical methods of like particle tracing velocimetry, or particle image velocimetry, which is based on cross correlation. Next slide. So those are very important, especially if you would like to see the how a particular cardiac device is working. So many times in the preclinical stages, when they are designing a new heart valve, they want to see how does that work. And so this is one of the images that shows how we use the particle image velocimetry, or PIV, to map the flow field downstream of a mitral valve prosthetic. Next slide. But there's a problem to that. And the use of laser, you can only do that in vitro or in a experimental lab setting. You cannot shine laser to the human heart, first of all, because it's dangerous. And second is that your heart is not transparent. So basically, the best way to do that is either to use ultrasound or to use MRI. So MRI is very expensive and it's also very time consuming. If you want to get the velocity field from a heart, you need to have your patient in a scanner. You need to have a scanner, first of all, and then it takes over an hour to get that data. And then because of the nature of the MRI, you can get just a phased average data. It's kind of averaging over a few different heartbeats. So one of our goal has been to develop, basically use this velocimetry technique that's been used with laser, now with MRI, with echocardiography. And we call this echo-PIV. So in collaboration with Professor Gianni Pedrizzetti in the University of Trieste, we have been working on these techniques for over a few years now. And so these are showing some of the 2D particle image velocimetry. And then the next step, which we are currently working, and we recently received a large NIH grant, is to be able to capture these velocity field in 3 dimensions. And this is very, very difficult to go from the 2-dimensional to 3-dimensional, because due to the physics of the acoustics, if you're going to have a 3D echo, your time resolution and spatial resolution are going to be much more limited compared to the 2-dimensional ultrasound. But, this is a technique that we have been very successful, and currently we are debugging the very final version of the software. And we are now hoping to look into a big clinical study with the help of the Cedars-Sinai Hospital in Los Angeles, that they are going to recruit patients with pulmonary arterial hypertension, and we are going to use this 3D echo PIV to assess that. The nice thing about that is that those patients right now, in order to diagnose and follow them up, you need to do a right heart catheterization. Right heart catheterization requires to send the catheter inside the heart. It's invasive, it's expensive, and it requires a lot of preparation for the patient. If we can do that through the echo, it just can be done in an office, should be very easily done. And that's our goal, to be able to minimize the suffering of the patients and lowering the cost of the healthcare by developing modalities that are non-invasive or minimally invasive compared to the current state of the art. You go to the next slide. This is showing one of the examples that how we use the three-dimensional echocardiography to kind of capture the velocity field inside the heart. This is actually one of the areas that I'm very interested to work with you all. One of the, obviously, you guys as computer scientists, you know that if you do have a very good system that works and compute things, in order for that system to be used in healthcare industry, you need to develop a graphical user interface that is easy to use and that can be used not just by the scientists, but also by the physicians. Obviously, to calculate these velocities, we do have like a high-end workstations that, you know, are a state-of-the-art, but not in every hospital they do have that. So you need to run those through the cloud system. So basically an interface will be in the hospital and they can easily connect to that, do the calculation, and also the system should be safe, secure, to make sure that the patient data is secured and is also compliant with all the HIPAA and all the local laws that protect the patient data. So these are the parts that I would love to seek your help and advice, and you know, if we can work together to make these things more accessible to the physicians and for the benefit of the patients. So yeah, those are basically, I started with our imaging projects that I know that there are some interest, and based on some conversation that I had with some of you, wanted to pose those type of projects to kind of work together. But I do have, I want to show you some other projects that are related to our efforts in medical devices. So this heart valve that you see is called A valve, or it comes from atrioventricular valve. It's the only modular bio-inspired transcatheter mitral valve technology. So currently, there is no transcatheter mitral valve technology that has been FDA approved. There are some that has been developed and are currently in the clinical trials. Each one of them has some advantages, has some problems. But one of the major problems that we have in the mitral valve disease is that there are multiple forms of diseases. And then one device generally cannot address every diseases. The reason for that is that you may have patients that they do have different sizes, for example, in their left atrium, different sizes of the left ventricle, and different sizes of the mitral valve annulus. And then you need to have a system that be able to accommodate all. So obviously, because there's no one system, we came with this idea of how to make a system that is modularly built. So in the cath lab, the physician will be able to say, hey, I'm going to use the atrial cage of size 28, and then I'm going to use a heart valve of a 26, and then combine them together easily to accommodate every patient's sizes and geometries. This is very important also with the different sexes. You know, men and women have very different heart anatomy. And other than that, the hormonal effect can come with different physiology. So with having those in mind, this is one of the work that we have been developing. So as you can see, it comes with different components. You can pick a bileaflet valve design or a trileaflet valve design. You will be able to kind of mix and match with different atrial cages. So one of those things that we did is obviously through the design of these studies, these devices, you need to check those. You need to check every details of those. So for example, here we were trying to see how does the effect of the height of the leaflet impacts the flow inside the heart. So we put two different bioprosthetic mitral valves that are designed and developed in our lab. We basically try to look into how the flow are different. So as you can see in the longer leaflet, it generates a vortex flow that is like a asymmetric. But when you look into the right one, which is the shorter valve, you see a symmetric vortex going downstream the valve. If you go to the next slide, please. So those are, I just showed it to you to show you how the imaging and devices sometimes help with each other. Those data were captured by laser PIV in a model that we have in the heart. This is a trileaflet version of the A valve that we have. Next slide, please. We put that, in order to put those valves inside the heart, you need to have a delivery system. You cannot just kind of, because when you do it transcatheter, it's avoiding the full open heart surgery that the patient needs to be on a bypass machine. So this is going to be much easier for the patients. They can go back to work much earlier than, you know, a complete, a full open heart surgery. So this is showing our delivery system and how we did that transapically. So this is a very short movie. It shows how the like A valve system works. It's basically considered minimally invasive. That goes through transapical insertion. So the system allows the repositioning of the valve, because when you're going there, you may kind of need to change some position. It gives the physician that capability. Next slide, please. So we tested that in the animal. These are done with in sheep. Go next slide. So you can see how the A valve nicely works, and there is no leak in the animal. So these are the process of preclinical testing that you make sure that your device is doing well. And many times through these experiments, you learn things that how to change a little bit in order to kind of improve your design and then move forward. One of the things that I was personally concerned is that because we do have an atrial component in these devices, I wanted to make sure that the atrial contraction is conserved. And this data actually showed that the atrial contraction is conserved and fine. So that's the A valve that we're still working on it, and we're hoping to do some long-term chronic animal study for the to kind of make sure that they are good valves to go for the next steps. The next project that we have been working on is Folder valve. Folder valve is a transcatheter aortic valve. So that replaces the aortic valve. The transcatheter heart valves generally started in early 2000. There were two valves. The one on the left is a pulmonary valve that was developed by Dr. Bonhoeffer in Great Ormond Street in London. And then the one on the right was the heart valve that was developed at that time by a company, PVT, and it was implanted by Dr. Kheradvar in Paris. So the one on the left is now commercialized by Medtronic, and it's called Melody valve. This is the solution for children who are born with pulmonary valve stenosis. And the one on the right was acquired by Edwards Life Sciences. Its third version is called Sapien 3, and this is one of the very good valves that are available. The transcatheter heart valves are very much adopting in the community, and even right now, most of the procedures are done through transcatheter routes rather than surgical routes. So this is much better for the patients because the procedure takes about half an hour, and then the patient go back home, and the day after he or she can go to their work, and there is no need for them to be hospitalized. Versus the previous version, which was like a surgical aortic valve, the patient should have been on the pump, and there were a lot of issues. But obviously, the problems are not completely solved, but there are still some issues. But overall, this is a major step in the human heart disease. So these are the current devices and older generation of the heart valves. So these three that are on the left are the ones that are currently approved in the US market. And they do have advantages and disadvantages, as I mentioned to you. One of the things that all these valves do is that in order for them to be small enough to be delivered inside the heart, they has to be crimped under a stent. And so these leaflets that you do have, mainly made of bovine pericardium or porcine pericardium, they are very delicate tissues that originally comes around the heart, which is the pericardial layer of the heart. And when they crimp that under these stents, obviously, the Laplace rule that if you do have a lot of stress, a lot of force happening over a very tiny area, which is like those stent straws, you're generating a huge level of stress to these tissue leaflets, which are very delicate. They're around 0.2 millimeter thickness. So as you can see on that image on the left, these marks are showing how the stent is basically stamped on it. And these may have some issues. One problem is the stent size, because obviously you can't crimp over something because of the conservation of mass. You do have some mass there. The other problem is that damaging the tissue, as shown in the left image. Next slide, please. So we have been able to characterize if you crimp those aggressively, you will get damage to the surface and the thickness of the tissue, mainly at the collagen level. that are not reversible. And that will generate durability problem. So obviously, when you have a patient that they would like to go for a transcatheter heart valve, they want to make sure that the valve that they're going to have is durable enough. You cannot do this every 5 years. For surgical heart valve, we know that the durability is at least 15 years and goes up to 25 years. We want to make sure that the same thing will happen for the transcatheter heart valve. The image on the left shows a natural calcification of an aortic valve, a native aortic valve, and as you can see, it's only happening on the aortic side of it. But the transcatheter heart valve, when they calcify very differently, and they calcify at both sides. And my hypothesis has been that these calcifications happen because of the damage that the stent crimping generates. Next slide, please. So overall, the TAVR or transcatheter aortic valve replacement short-term and mid-term clinical outcomes are really good. Next. But the limitation that the clinical trials are showing is that when they look at them, for example, in 8 years, they see that after 8 years, only 10% of the patients are alive. So 90% died, so we don't know what happened to them. But in that 10%, they say, oh, these 10% who are survived, they are doing good, okay? And then there are 3 different stages of durability, which I'm not gonna go over those, more technical, but I would be happy to show you those. So that's kind of a bias that existing in the analyzing the data for a long-term TAVR durability. So a structural valve deterioration is the technical term for the durability, has been basically reported only according to reintervention. So, for example, if you do have a patient that has a problem, like a so severe problem that needs to have his transcatheter heart valve replaced again, then they count it as this is one failure. But a lot of these patients are too sick or too old that they cannot go over another procedure. So for those, it was not counted. Very recently, the guideline has changed in order to be able to more characterize those. But still, the survival bias exists, meaning that a clinical trial starts with 100 patients. In 8 years, there are only 10 patients available. So what happened to that 90 patients? Whatever results you're going to get in that 10 patients is not reliable because of that bias. So we come with a new technology to kind of address that issue of crimping. So fold-a-valve is a transcatheter aortic valve system that uses a very different mechanism for the folding or for crimping. Basically, the leaflets are going to be crimped completely outside the stent. So you do not crunch them through the stent. And then the fold-a-valve technology also has some additional advantages. For example, it is the only transcatheter heart valve that is repositionable. It is retrievable. So if a physician goes there, something goes wrong for the patient, they can take it back still without opening the whole heart. So that's going to make it much easier, much better for the patient, and much cheaper, because right now, for every transcatheter procedure, you need to have one interventional cardiologist and one surgeon. Most of the time, the interventional cardiologist does that without any problem. but the surgeon should be there in case that there's a problem. The surgeon opens it and take care of it invasively. So assume that you do have a system that's more reliable, that if there's a mistake, you can take it back non-invasively. So you probably do not need a surgeon in the room. So overall, the cost of procedure is going to be much lower. So this is kind of showing how the Folder valve works, basically when it's inside the heart. This is one of the versions of Folder valve that we have. We developed it in our lab. Next slide, please. So this is kind of showing how does the Folder valve crimp compared to the classic crimping. Thank you very much. So the classic crimping, the leaflets are inside the stent. So it not only avoids the injury to the tissue, but it also can reduces the profile. It can make it, because now you do not put everything inside the stent. You have it outside the stent, so you will be able to even crimp more, because the smallest that you go, you can use it for more patients. A lot of elderly patients, they have very fragile vessel. You cannot go with many of these current technology because they're too big for them. Or for younger children, you cannot go because their vascular system is very small and tiny. So you need, as you can go smaller, the better. But with the current technology, if you go too small, then you basically fully damage the tissue. So that's kind of an optimization problem. So Folder valve is basically smaller than all the current available valves in the market. Next slide. So this is kind of showing is like how the Folder valve things work. So Folder valve is going first, it's inside and then coming outside. And first is outside and then coming inside and then form the valve. And this is the way that we can take it back. We can completely take it back. But if you look at it on the left, these are how the, like for example, Edwards Life Science Sapien is crimping. And as you can see, that delicate tissue is completely crunched within the stent on the sides. Next slide, please. So we also have done a lot of imaging to see how the flow changes. Like for example, this is flow downstream the Folder valve. This is using laser PIV. This is 1,000 frames per second. And you can see the vortices in the sinuses of Valsalva. You see how the flow is rotating, meaning that this is really good for the oxygen to come back to the coronary system. Next slide. This is showing how the Folder valve delivery system works. Developing these delivery systems are really, really challenging. They are it's like a finest watchmakers type of a work. And I am fortunate that one of our team members, Greg Kelly, is a very experienced engineer in developing these delivery systems. This is showing that how we can compress and expand. So these, we call it a staged deployment, allows the physicians to kind of work on it, make sure that the valve is sitting in the right place exactly, and tested everything before going out of the patient. So that allows a much better performance for the physician and for the patient. Through these studies, you have to make sure, and this is the FDA mandate, you have to make sure that these valves can survive 200 million cycles. And that's usually around 5 years in a heart. And so there are machines that developed for that. They called accelerated wear test. So these are showing Folder valve in one of these accelerated wear tests through or kind of preclinical studies to make sure the valve is good. After 200 million cycles, we look at them under microscope. We want to make sure that all the components are still fine. There is no damage to the stent. There is no damage to the fibers. These are very, very important. This is showing the scanning electron microscopy of the Foldo valve. So you see how smooth these holes are, because a little bit of a friction can generate like a torn of those fibers that they are used there. So it requires a very meticulous job in designing those. This is an animal study that we performed in Paris with Foldo valve. So it shows how the Foldo valve is implanted inside the sheep heart. This is under fluoroscopy, and this is basically the standard way that they do the transcatheter heart valve. It's always under fluoroscopy, even in the patient. So you see that the physician can work on it, make sure that the coronary arteries are open, because if your device is occluding the coronary arteries, the patient immediately goes under a myocardial infarction and will die. So this is very, very important. So this is like our physician is now trying to reposition the valve, make sure that it is positioning in an optimal position before it goes out. So as you can see, it's a heart beating. The animal heart is beating. Now we see to make sure that the coronaries are completely open and it's not obstructing the coronary arteries. And now we are releasing the valve fully and we go out. So again, we want to make sure everything is working well, and that this is kind of a dye that you inject to make sure that the coronary arteries are open. This is showing the sizing after when we implanted, and everything is kind of as planned. This is again fluoroscopy. This is the sheep heart. And obviously there are two designs, so we learn a lot of information through our design, through our animal studies, like the newer design are a little bit shorter than the older design. If you go to the next slide, please. Yeah. So these are the echocardiographic study. We want to make sure that the valve is working perfectly when it's implanted in the animal. Next slide. And then more recently, we have even advanced our Foldo valve, and we have incorporated for the first time an intravascular ultrasound technology inside of it. So that will allow not only an Eulerian view, which basically what you see in through the fluoroscopy, but it also gives you a Lagrangian view. It's like over the catheter. It's like when you're driving your car, you do have a Lagrangian view of the street. And if the police that is standing, when they see the cars are moving, they have a Eulerian view. So with this, we are providing both Lagrangian and Eulerian view to the physician who's putting it together. This is the new delivery, the handle of Foldo valve, and that red thing is the tip of the IVUS catheter. So this is kind of showing how the IVUS shows the Lagrangian view when you are advancing into the vasculature, and then the other one shows how you will be able to capture the calcification through this. And this is unique because through the angiography, you cannot see the native valve calcification. So this is the new Foldo valve. We have recently also got a new grant to kind of use that for longer term chronic studies. Next slide, please. So overall, fold valve should improve the durability. It avoids vascular complication because it's very small. It should eliminate malpositioning because it has the option to reposition, and it can improve the navigation and deployment through its IVES system and minimizes the paravalvular leak because of the skirt system that it has. Next slide, please. So that was fold valve. And then another recent technology that we are developing is called Helix Cardia. Helix Cardia is a heart support system. Right now, for the patient with end-stage heart failure, there are only two options. Either they're lucky enough to receive a heart transplant or ventricular assist devices. So they're currently in the US between 150,000 to 250,000 end-stage heart failure patients, and there are only 2,500 hearts per year to be transplanted, and about 2,500 ventricular assist implants. There's a huge gap only in the US. I do not have the statistics for Mexico. I actually looked for it to kind of report to you, but I couldn't find it. But I'm sure a similar situation exists in Mexico as well. So the current VADs basically change your pulsatile type of pulse. Your pulse changes into a continuous flow. So you will lose the pulse. And obviously there would be a lot of problem with respect to the losing the pulse in the heart. As they said, VADs extend the life, but viewed with huge loss of quality of life. So we developed a Helix Cardia according to the helical heart fiber arrangement, and that's a discovery that by a Spanish scientist, Francisco Torrent Guas, who he showed that the heart actually is a band that is helically arranged. So we developed something like that. Next slide, please. And that's Helix Cardia. So Helix Cardia is a minimally invasive cardiac support system that wraps around the heart and is minimally invasively implantable and is wirelessly, transcutaneously rechargeable. So it can be used for patients who cannot undergo like a heart transplant, or they prefer not to receive the VADs. So this is kind of show how the ejection fraction calculates according to the design, the way that you design it, and the angles that you're considering. Next slide. And it's controlled by a pacemaker system. Almost all of these patients with heart failure, they do have a pacemaker, so that can be used for that. And this is kind of, obviously we are at the moment in the surgically.
And that is, I'm going to say,
this is showing the peak heart, and we have been developing, currently testing those. If you can play those two echoes. No, if you go back, there are two movies. Yeah. So we induce the heart, we basically induce a heart failure in these animals, and we compare. So if you play the top one, that is showing the heart that's kind of induced with induced heart failure. you see that it's not really working. And then the second one, it basically when the device is on, so you will see the same when it's been assisted by our device. And if you go to the next slide, we have been able to add 32% to the ejection fraction of the heart. And that's huge because a patient with a 10% or 12%, like with an ejection fraction of 12%, which is very close to death, can have a 44%, which is something that they can live with. A normal heart is between 45 to 60 ejection fraction. We also have shown that there has been no adverse effect on the epicardium after you take the device off. So yeah, that's kind of, this is my last project that I'm very excited to show you. It's called mitochondrial transplantation. So we were able to transplant the mitochondria from different species and different cells into different cardiomyocytes. And we showed that the bioenergetics of the cell can be supercharged and can be improved. This is very important with respect to helping patients who are born with mitochondrial disorders and congenital cardiomyopathies. And generally, this idea of mitochondrial transplantation is believed that can be even used for rejuvenation of the humans in different things. Obviously, we are only working on the heart part of it, but there are other scientists that are working on other things. And it's been, one theory is that the reason that we age is because of the mitochondria that we have in our cells. So at the end, I would like to thank the funding agencies who funded our work through the years, and also several of my good trainees and colleagues and collaborators that, I put just some of them that are mostly involved in the projects that I discussed, and I may have missed some. So these works are done, it's not a one-man show or one-man work. These are done through collaborations through the years. And that's something that I always enjoy, their collaboration with industry, collaboration with international collaboration with scientists, with physicians. So that's the way that we can be effective and efficient and do something that at the end help the patient. And at the last, I would like to acknowledge Leonardo da Vinci, who is the very first cardiovascular engineer. And this is my very first project in this. When I was a PhD student, my PhD advisor, Professor Maury Garib in Caltech, asked me to make a prototype similar to the Da Vinci sketches, because in 1500, he expected that the flow inside the aortic valve has these vortices. So according to his design, I developed that heart valve, and then we did the particle image velocimetry to kind of see how does the flow look like. This is exactly matching what the genius Leonardo predicted. We don't know how he did it, that he imagined the flow will be like a vertical. Anyways, this was even shown into the Victoria and Albert Museum in London multiple times through the experiment design of Leonardo da Vinci. Thank you so much. I hope I wasn't too long for my presentation. Thank you for your patience. Appreciate it.
Now it's time for questions and answers. So Yeah. Very much. Your name and the question.
Thank you. Hi, thank you for your presentation. My name is Magdalena Saldaña. So I have two questions. The first one is, of which material are done the frames of the valves? Because you said that the membrane was done of pork and bovine, but the metal is, what kind of metal do you use?
Very good question. So the one that I showed you from our laboratory are made of nitinol. Nitinol is a alloy of nickel and titanium, and it has it is self-expandable. So basically, it's a shape memory, and it is very elastic, basically. There are some others that if you do through a balloon expansion, they usually use cobalt, chromium, or a stainless steel. But the one that I showed you from our lab are self-expandable, made of nitrogen.
Okay, thank you. And the other one is, I see that you have a lot of projects, so it's a lot of work. But in your lab, do you work by little cells, by little groups, or how do you go over all these projects at the same time?
Absolutely. So everybody in my lab has a different project. So like we do have a small group of people who are working on a particular project. So for example, the ones who are working on the cell culture, they're completely different than the groups that are working on the deep learning. The ones who do like a computer programming related to PIV or echo PIV are very, very different than the ones who work on the prototyping. So we do have different teams that are working on different projects.
Thank you very much.
Thank you.
Some other questions? Yes. Please, your name and the question, please.
Thank you very much for your presentation. My name is Blanca. And my first question is about the replacement of the mitral valve. What happens with the native valve?
Again, very good question. So mitral valve usually replaces for two reasons. One, which is more common internationally, is due to rheumatic heart disease. And the other one, which is more common in the US and in Europe, is basically due to the a leaky mitral valve. So when you transcatheterly replace those, you do not really replace them. You implant a new valve inside the old valve. So you still keep the old valve there. But when they do surgery, they actually cut the one and kind of rechange it. But yeah, to answer your question is that the native valve stays inside the valve.
And another question, you mentioned that you have bicuspid and tricuspid valves. The mitral valve is bicuspid. So the tricuspid is for the valve.
Again, very, very good question. So we do have four heart valves, and among those, mitral valve is the only valve which is bileaflet. And you ask me why, I have to say, I don't know. Nobody knows why, like an evolution kind of decided the mitral valve to be bileaflet. There are some hypotheses, but none of them really, you could not prove any of those. But there are some reasons behind that. But in the industry, almost all the valves that they develop to replace a bileaflet mitral valve still is a trileaflet. There are several reasons for that, because when you do have a trileaflet valve, when the valve is open, is completely circular. And from the engineering perspective, to make it is much easier to make it that way. When you do have a bileaflet valve, you need to make sure that the annulus of the valve is kind of deflecting, similar to the native valve. Because if you make something that has a rigid annulus, and if you have a bileaflet valve, the way it opens is like this. It's like an eye. It's going to be still a small. So that's kind of the way that they do it. One of the valves that we have developed, the one that I showed you, the bileaflet one, is it has a dynamic annulus. It has a nitinol base that can deflect back and forth. So it opens up enough to kind of generate the flow required.
And what happens with the sound? How does it sound? Because I remember I heard once a valve that was metallic, and it was a click, click, click. How is this noise?
Those are the old mechanical valves that they had a click. These are bioprosthetic valves. These are made of like tissue. So the tissue, they do not have any click.
So the first and the second heart sound do not change in sound?
No, not really.
If you listen to a stethoscope, is the same?
It's not exactly the same, but you don't like, the old ones, the mechanical ones, one of the problems that the partner of the spouse of the patient has is that, doc, I can't sleep because, my husband or my wife clicks at night. So that was the old problem. But the new mechanical ones, they do not kind of have those sounds. But the bioprosthetic one, obviously the sounds is a little bit different if you really look into it, but it's not something that bothers the patients as it used to with the mechanical.
And it doesn't produces any extra sound like a murmur?
No, murmur usually happens if you do have a leak. If they are correctly there, if there's no paravalvular leak, you shouldn't have any murmur, like a specific murmur. But the magnitude of the S1, S2 that you have in a natural valve, they are a little bit different if you listen to those.
Okay, and my last question. What happens with the viscosity of the blood with these valves? Does it affect? Like in
viscosity
anemia?
of the blood. If you do have mechanical valves, you definitely need to have a blood thinner for the patient because those surfaces are not biocompatible enough. So there would be hemolysis. So the patient needs to be on blood thinner. For the bioprosthetic valves, until 2017, they said you don't need any blood thinners, which was actually great because these blood thinners have a lot of problem for the patients. But then in 2017, some studies showed that there is a concept which is called subclinical leaflet thrombosis. So there's a thrombosis that generates over the leaflet. And it's interesting that these are happening much more often in the transcatheter valve compared to surgical valve. And again, my theory is that because of the damage due to a stent crimping over the leaflets, the platelets can deposit easier and then generate some kind of a gooey form thrombosis there. But for those patients, for some limited time, they start to have blood thinners to make sure that those subclinical leaflet thrombosis won't happen because they can be potentially very dangerous because if one of these thrombosis kind of detach from the leaflet, it may end up into the brain and it generates a stroke.
Thank you very much.
Thank you very much.
Anybody else? Dr. Shima, please. It's your time.
Thank you very much for your wonderful talk. I have two questions. I would like to know what happened with the immune system. Do you have troubles with your immune system when you put these valves in the heart?
The valves are, so the tissue are glutaraldehyde fixed. So they take like these leaflets before they're gonna be used for the heart valves. They're de-antigenized. So they do not have the antigens, but because like obviously they try to remove all the nucleus or whatever things that like antigens from the pig or the cow, but this is always 99%. There's always a 1%. So one of the reasons that we do have calcification in these valves is because the immune system kind of tried. But overall, like in the 70s, which the tissue processing was not as great and they did not know too much, we would get the calcification super fast. But nowadays, it is much less. And then still, it's interesting that they don't put that into the children. Children or youngs, anyone younger than 40, they don't put it because these valves calcify faster. So I guess one of the reasons could be due to the like immunogenic response that are more are higher in the younger compared to the older.
And my second question is related with the data in order to validate all your computational things that you did. Do you have troubles with the data in order to validate it with the medicines? They give you the data, the correct data?
The deep learning? Yeah. Yes, obviously, you know, we do. I collaborate with UCLA, with USC, with Children's Hospital Los Angeles, with Heart Center in Hamburg. You know, I work with so many different institutions in the US and around the world. But generally, as you know, for these training, like for example, if the way that Facebook kind of understand who is who, you know, they do have billions of data for training. In the cardiac, with the way that we can work on it, at most is thousands, and you know, it's even not easy to get thousands. And obviously, the complexity of the medical images is much more than just the face of a human. So overall, in order to be able to do a job that Facebook does, you need to have millions of data, and it's impossible to get that.
Thank you very much.
Thank you very much.
Someone else? Yes, Dr. Rivero. Mario.
Thank you so much. Thank you so much for your talk. Most of the studies that you presented are made for evaluation in like clinical environments or control somehow environments. Are you interested or planning or working in ambulatory studies for the heart disease? Like, for example, high performance athletes have like sudden heart problems in the middle of the game, something like that. Is that in your range of research?
Absolutely. So one of the concepts that I did not really discuss here is the concept of vortex formation time index. That's an index that we developed and show that it has a relationship with respect to the diastolic function. So one of the projects that I always wanted to do that is to test those in the athletes, and it requires just the ultrasound, just a simple ultrasound. But it never happened that, kind of I get access to kind of that type of a data. But yeah, those are all, I think that's a unique study, nobody, to the best of my knowledge, have done that.
Thank you so much.
Thank you, Rocio.
Thank you very much. I would like to know about the funding. I mean, are you working with the industry or is just a project of the university?
No, university, you have just usually very small amount of funding that, cannot cover all those. Most of my funding coming from the NIH, from NSF, from American Heart Association, and I do work with industry as well. Some of our funding comes from industry.
Okay. Yeah. Thank you very much.
Thank you.
Thank you, Rocio. Somebody else? Okay, let's give a big applause to Dr. Arash Kheradvar. Thank you very much.
Thank you
for
so
this
much.
meeting. So I might ask, Doctora, could you please, the diploma of this event for Dr. Arash? Arash. It's not so easy to pronounce many words.
Thank you so much. Thank you. Thank you.
You can read it in Spanish.
Okay, on the next step, we will take a photograph of this event today. So I will please invite you to go outside for a
Sure.
photograph. Thank you.